In this set of slides, I want to discuss some of the mechanisms by which enzymes' activities get regulated. This is not an exhaustive list, but I think this is the most important set for you to become familiar with, some of which are going to be fairly familiar. For example, allosteric regulation of protein function and enzyme activity is something that we've discussed in previous lectures. Some of them might be a little bit new. In any case, we're going to run through some of these examples, and then we'll see in class more application of these in specific examples. Let's begin with protease activation. In the case of the enteric proteases, we have a whole suite of serine proteases with slightly different target molecules, slightly different substrate sequences or substrate specificity, and however, these proteases exist as inactive zymogens secreted from the pancreas into the intestines. In this example, we're looking at chymotrypsin, which is here the final active state of the molecule. Remember, serine protease, here's our catalytic triad, and the color coding in this slide just shows us the different independent polypeptide chains that exist after proteolytic digestion of the zymogen. So here we see 245 residue single polypeptide chain, chymotrypsinogen, but we see that that chain is degraded or digested by trypsin, and first into 2 polypeptide chains and then into 3 with the loss of a couple of dipeptides. So we're not losing a lot of sequence information, but the overall structural information is changing just because we no longer have one contiguous chain. Now the chain, I should say, is still held together through the use of disulfide bridges. You can see those in a few different places in this cartoon. The point is, though, that the chymotrypsinogen exists as a reservoir of inactive protein that can be readily activated. So what's the signal? Well, we're not going to discuss the signaling mechanism in detail, but suffice it to say that upon ingestion of food, enteropeptidase is secreted from the small intestine, and enteropeptidase is sort of the keystone activating a set of other molecules that are already extant in the intestine, again, in their inactive zymogen state. So trypsinogen, for example, is cleaved by enteropeptidase and by its own active form, so we autoactivation as well as activation by enteropeptidase, and then we see that trypsin goes on to activate, as I mentioned before, chymotrypsinogen as well as proelastase. So the key here is that we're getting an amplification of whatever signal we're receiving at the front end by perhaps many orders of magnitude, so that we can go from a state where we're not degrading protein to one in which we're actively going to degrade protein with this whole suite of protease enzymes. Something I'm not talking about on this slide is, of course, the negative regulation of these enzymes. What you'd say, well, okay, Gillespie, we've activated all these enzymes. What happens when we need to shut them down? This is an irreversible process. We have no way of going back to the inactive state, chymotrypsinogen or what have you, to the zymogen state. So what do? Well, we have another set of proteins. These are the inhibitor molecules that are protein-based inhibitors, and they have a very low KD, very tight binding to their target enzymes, and so we can readily shut down the activity of these proteins in response to a different signal. Again, we're going to skip over what the signals are in this case. Here's another example of an activation cascade. This is a blood clotting cascade, and I'll say right away that many of these steps involve the serine protease-based activation of a target molecule. These are proteins that circulate freely in the bloodstream as zymogens again, but they're activated in most cases by some serine protease activation step. I'm not going to go into great detail. Suffice it to say that we have 2 different pathways in which we can respond with the formation of a blood clot, and those 2 pathways converge at thrombin. And there's a couple of details to point out to you, again reminding you that in each of these steps mostly what we're talking about is the activation of a zymogen to its active state, and in most cases that involves again the activation of the zymogen to form an active enzyme that then goes and acts on a subsequent target. A couple of interesting things to point out here. Again, I'm not going into all of the details in describing this, that's not my objective, but one thing to point out is that there are elements of this pathway that are subject to inactivation as well. And I mentioned protease inhibitors, intrinsic protease inhibitors before. Here's an inhibitor that inactivates the conversion of factor 7 to factor 7A. As well, there's antithrombin that acts in a couple of different ways to inactivate the thrombin activation, part of this cascade. But ultimately, again, what we are shooting for is the activation of prothrombin, and thrombin then can go on to convert fibrinogen to fibrin, and then we get down to our formation of a blood clot. The other thing that's worth pointing out about this guy is that it's of molecular biological importance. The activity of thrombin is of importance to the science of molecular biology, in that, as you know from our previous studies of serine proteases, this molecule is a protease that acts on a particular sequence. It has a substrate specificity for this sequence. It cleaves the peptide bond between these two amino acids in fibrinogen, and therefore we can use that as a means of assisting our protein purification efforts by linking our target protein, our protein of interest, via this thrombage cleavage site to another protein that's easy to purify, for example. It might be an antibody. It could be any number of different things. The point is that we can tether our protein of interest to some other easily purified molecule, express it, and then begin to purify it. And then once we've gotten our pure target molecule, we use thrombin to cleave this linker sequence, and then we can retrieve our pure protein and purify away the excess baggage. So that's another reason to show you this activation of zymogen pathway. But let's move on to another kind of regulation of enzyme activity. That would be via reversible covalent modification. Obviously, protease-based activation is an irreversible process, as I've suggested a couple of times already. But here are a few different players in the reversible modification of proteins that result in activation or in many cases too an inactivation of an enzyme. And again, this is not an exhaustive list, but this is a series of modifiers that you should be well aware of: phosphorylation, acetylation, acylation, ubiquitination, which we've already discussed in other cases as being the addition of one or more ubiquitin monomers, right? That's a relatively large protein that can signal degradation as well as targeting the labeled molecule or the modified molecule to other pathways, and then glycosylation, which we're going to encounter in the last week of the class. Here are the molecules that are donated by these. I'll point out acetyl-CoA as being of particular interest given the reader article that I've given you to think about. But the other thing that let me point out here is two things. First of all, acylation, ubiquitination, and glycosylation, these are often involved in targeting of the protein to some specific location. Phosphorylation and acetylation are two processes that are more involved in the activation or inactivation of the enzyme. And it's worth pointing out that the donors in these cases are useful indicators of the metabolic state of the system. ATP, of course, being the energy currency, but of course acetyl-CoA being just as important in determining the metabolic state of the system. So you can imagine that it will be useful to use these very molecules that tell on the metabolic state of the system to actually modify the enzymes involved. And we'll see that when we look at our reader article in class. Just pointing out, too, though, that the usual suspects are involved in accepting these functional groups. They're polar side chains that we often find modified. What I'll also point out too, which I'm not going to go into much more detail in, is the idea that not only do these amino acids accept the functional groups in the modification process, but there's more than that. There's the rest of the sequence around that acceptor site, and this can be very specific sequence dependence in terms of what proteins get modified by what other molecules. And again, this is something that we'll explore in greater detail in class. But it should give you the sense that we have very fine control over the modification of a particular protein based in part on the sequence element that's governing the acceptance of these modifications. Now this slide is just going into a little bit more detail on phosphorylation. There are many eukaryotic kinases. We know that kinases are enzymes that add a phosphoryl group to other molecules. So these enzymes then are the activators. They activate other molecules through reversible modification through the addition of a phosphoryl group to a serine, threonine, or tyrosine most of the time. And the key to all of these changes is, of course, with a new functional group, we have new structural information, new chemical information that drives structural changes that alter the activity of the protein and also perhaps alter the affinity of the protein for its other target molecules, be they small molecules or be they protein-binding targets. Another interesting idea here is that the length of time that we induce the modification in enzyme activity is dependent on how stable the modification is. I told you these are reversible changes in structure, and of course the reversal drives us back into our initial state. So there's two ways that this can happen. One could simply be the auto hydrolysis of in this case the phosphoryl group, or there might be an enzyme assisted mechanism by which we remove that phosphoryl group and then again driving us back into the initial state, whether it's the active state or the inactive state. We see both of those. We see both activation and inactivation of enzymes by phosphorylation. So again, pointing out that the kinase is the enzyme that adds the phosphoryl group, and the phosphatase is the enzyme set that removes the phosphoryl group. Again, the idea being that not only do we want to not only might we want to activate a protein, but we'll want to inactivate the protein as well. Again, the key being that we respond to the needs of the system at any given moment. It's interesting to point out that, though I'll dwell on this for another second, the time-dependent change in the time-dependent hydrolysis of, say, a phosphoryl group is really interesting because this is an intrinsic characteristic of the phosphorylated protein, and you can imagine engineering a system that will respond quickly or slowly to phosphorylation. So we can essentially modulate how long we respond to a signal or a stimulus in the organism by how long we retain that phosphoryl group. Okay, we'll come back to some covalent or some reversible modifications, but let's turn now to allosteric regulation of enzymes. This cartoon is one that I've shown you before. What we like to assume is that we have this one way to think about it in any case is that we have this pre-existing equilibrium between two conformations. One is the tense, that is in this case the inactive state. One is the relaxed, that is the active state. And that we can drive this equilibrium backwards and forwards through the presence of non-covalently bound ligand molecules. So in this case we see that this ligand, this might be a positive allosteric effector driving the equilibrium towards the relaxed state. So conformational change is often affected by downstream or upstream metabolites. So in this case, let's consider that the system that we're regulating, the enzyme that we're regulating, is part of a metabolic pathway, and that this enzyme that's part of that pathway responds to allosteric effector molecules. This again is something that we'll discuss a little more in class, but the interesting notion here is that it's the metabolites themselves that can be the allosteric effector, and that makes a certain amount of logic to imagine that the activity of a metabolic pathway is going to respond to concentration changes in either precursors or final products of the metabolic pathway. Another key here is that this is a non-covalent association with the target, although we've seen that idea of non-covalent versus covalent interactions in allostery breaks down a little bit. I'll point out the Bohr effect to you and ask you whether CO2 or carbonic acid is an allosteric effector of hemoglobin, or whether the protons that bind to the tense state of hemoglobin and induce those tense state salt bridges are an allosteric mechanism, even if they are covalent changes. So it's a little bit blurry at this point here, but the big idea is that we see changes in the structure of the molecule that affect a change in the activity of the molecule. And we've talked about some of this, what allostery does to our rectangular hyperbola dependence of activity on substrate concentration. And I'll show you that again in another slide or two. Another detail here might be that the places that where the enzymes which we choose to regulate in our metabolic system tend to be the smart ones. That is to say, the places in a metabolic pathway that cost a lot of energy or in which we gain a lot of energy, and we'll elaborate that on that in another moment. But let's take as an example aspartate transcarbamylase. This is a complicated enzyme, and this is a silly cartoon of it here, so forgive me for the circles and boxes. But this is an enzyme involved in DNA biosynthesis, in particular pyrimidine biosynthesis. It's a useful model, however, because it shows an organization that's fairly standard. That is to say, we have a set of catalytic subunits. In this case, it looks like we have a dimer of trimers, where we have a trimer of catalytic subunits interacting with another trimer of catalytic subunits. But the regulatory subunits are quite distinct. So we're affecting the regulation of this enzyme through a different set of polypeptides that are non-covalently interacting with the catalytic domains. Of course, and here we have the red effector molecule, whatever that's going to wind up being, we'll see in another slide, that binds to those regulatory subunits, and you can imagine that binding event causes a conformational change that is transmitted to the catalytic subunits, and therefore we inactivate this enzyme upon binding of this allosteric effector molecule. So this is there's a couple of things to make sure you take away from this slide, is that oftentimes, not uniformly so, but oftentimes we see the separation of the regulatory function from the catalytic function. I think there are obvious benefits to that. I want you to chew on that and think about that, and we'll bring that to class. And then to that the places in a metabolic pathway that tend to be regulated are the committed steps, or the expensive steps, if you will, in that metabolic pathway. Here's another slide on that same enzyme, aspartate transcarbamylase. Again, this is an enzyme that uses aspartate as a substrate and accepts a carbamyl group from carbamyl phosphate, resulting in this precursor molecule. What's the point of this pathway? To make DNA, right? So here's a DNA biosynthetic precursor molecule, and this is a long pathway. This enzyme, ATCase1, is just the first of 7 different enzyme transformations that take place that result in the formation of this precursor molecule. Now, we've seen these red lines with little bars on them already in previous slides. Again, this is indicating where our particular enzyme, the transcarbamylase, is being downregulated. So these molecules, the endpoint of the metabolic pathway, is an allosteric effector that negatively regulates the activity of the first enzyme. And I think it should make sense to you why that's a logical way to regulate the enzyme. If the concentration of CTP is high, then there's no reason to continue creating these precursor molecules. So an abundance or high concentration of the endpoint tells us that we can shut down the pathway at the beginning, right? So we don't expend any more energy in creating these intermediates or the end product. So, and we also see that UTP, one step earlier in the pathway, also negatively regulates the enzyme. And this is where I drag these figures from, but I'm showing this one to you as well, just because this ties us back to our previous discussions of how we look at enzyme activity. And let's selectively use our attention here to see that in the presence of ATP, we have this rectangular hyperbola, and let's just accept that ATP is involved in the biosynthesis of the end product. So we see that as we increase aspartate concentration in the presence of ATP, we get a better and better and better activity, and we saturate at some point. But in the presence of the negative allosteric effector molecule, we see what appear? Well, it looks to be a sigmoidal dependence on aspartate. So now we see that we have negatively affected the activity overall, and we see that now the allostery in terms of the response to aspartate is enhanced here. So that is to say that In the presence of CTP, we have low activity, but then when the aspartate concentration increases, we can overcome that at extreme concentrations to get back to our high overall enzymatic activity. Should be all of these details should be familiar to us by now. Okay, now what else do I have to say about this? So this is a complicated idea, but let me read it to you and see if we can decipher it. Oops. Reversible covalent modification, which we've just been talking about a couple of slides ago, phosphorylation in this case by a kinase of target molecules, is itself under allosteric control. So frequently the point is that we will encounter multiple layers of regulation. Let's break it down. Protein kinases do what? They phosphorylate other molecules. The point of phosphorylating those target molecules is to activate or inactivate them. However, the kinase itself is under regulation, allosteric regulation. So this is protein kinase A. It is a dimer of dimers. There are two catalytic subunits here denoted in blue, and the two regulatory subunits here denoted in yellow. The regulatory subunits bind cyclic AMP. In non-covalent binding of cyclic AMP, we see that here in this blue cartoon. I'll come back to cyclic AMP in a moment, but suffice it to say that for now, that cyclic AMP binding at the regulatory subunit induces a conformational change that causes the release and subsequent activation of the catalytic domain. So upon dissociation from the regulatory subunit, the C subunit, the catalytic subunit, is active. How is this accomplished? Okay, two details here. Here's a model of cyclic AMP. This is a second messenger molecule. I'll talk about it again in a moment. But this molecule tells us that something has changed. There's some signal or stimulus has been received by the cell, and then a regulatory subunit senses that as a concentration increase of cyclic AMP. And then what happens is that there's an interaction between the catalytic subunit and the regulatory subunit that happens to be via the substrate binding site of the catalytic domain. So this is referred to as a pseudo-substrate sequence, the sequence being part of the regulatory domain. Basically, the regulatory domain mimics the natural substrates of the catalytic domain, which makes perfect sense, but that mimicry is dependent on the presence of the second messenger molecule. So the kinase, which is involved in covalent modification of its target molecules, is under allosteric control. I have a couple of slides that I want to show you before we leave this idea, because some of the background here might not be familiar to everyone. Okay, here's a nice cartoon. This is just stolen from Wikimedia Commons, involving the G-protein coupled receptors, and this slide is about the generation of cyclic AMP. So I mentioned that cyclic AMP is a second messenger molecule. What does that mean? It means that there's a first message that's received at some point by the cell. So what we're talking about here overall is the propagation of signals, signal transduction. And what we know is that the cell has to sense the changes in its environment and respond to those changes. So that's basically what we're talking about, right? When we're describing the regulation of enzyme activity, we're talking about the change in the state of the cell in response, the state of the enzyme in response to cellular conditions. So again, backing up, cyclic AMP is our second messenger. It's activated, it's converted to cyclic AMP from ATP by this enzyme, which is adenylate cyclase. Right? It's just an enzyme that cyclises ATP. Let's back up again and show you that the cyclization has to do with the phosphoryl group. There's the nucleotide base, there's the ribose, there's a phosphoryl group, and we note that this is an unusual arrangement. So this is the end point of the signaling process, and you can imagine now that this cyclic AMP is going to wander off now to bind to the regulatory subunit of protein kinase A, thereby activating the catalytic subunit, blah blah blah. But where does it come from? So here's a receptor molecule. It doesn't really matter what it is. It's in red. The hormone in this case is coming to bind at this 7-transmembrane alpha helical receptor molecule. The key though is that upon ligand binding, the receptor changes structure that allows it to interact with this G protein and exchange at the alpha subunit of the G protein a GTP for a GDP. Okay, so this is an interesting detail. I haven't. The idea here is that, please forgive me. The idea here is that this cytosolic G protein is a sensor of what's happening on the outside of the cell. It responds to a conformational change in the receptor. The conformational change in the receptor signals to this molecule that it needs to bind GTP and hydrolyze it. But before it hydrolyzes that GTP, now it's in its active state. This molecule, the alpha subunit of the G protein, can wander over to adenylate cyclase, right? You see now the G protein interacting with adenylate cyclase, telling adenylate cyclase it's time to generate cyclic AMP. And then there's a hydrolysis of GTP to GDP, and that results in the inactivation of the molecule again, and that hydrolysis resulting in the release of adenylate cyclase. So the key here is that it's complicated, which is why I wanted to give you this extra slide. We generate second messenger in a way that depends on the presence of some signal. That signaling is mediated through the G protein, and the response of the G protein to the signal depends on how long GTP is present. So I mentioned before autohydrolysis of a phosphoryl group in a phosphorylated protein. In this case, we're talking about a similar mechanism, sort of a self-timer, if you will, of how long the signal is received and transmitted to adenylate cyclase, that being based on how long it takes the G protein to hydrolyze GTP to GDP. Hopefully that's simple enough for you. But the bottom line is that in the end of that signaling process, we have generated our second messenger, cyclic AMP, which again allosterically changes the structure of the regulatory subunit, allowing us to uncouple from the catalytic subunits of protein kinase A so that it can go off and do its job, phosphorylating a whole host of other molecules. I think that allosteric regulation of protein kinase A is just a little bit too fun to leave right away, so I have a couple more slides to clarify this to you. In this cartoon, stolen from the Taylor group, you can look this paper up if you like, we've drawn the catalytic subunit as this sort of light beige, and the regulatory subunit or part of the regulatory subunit of protein kinase A in gold. And the key here is that there's a conformational change in the regulatory subunit upon binding cyclic AMP. And I've highlighted for you the cyclic AMP here. It's a little bit difficult to see, but this red circle shows you where cyclic AMP binds in the regulatory subunit, driving an enormous conformational change that is concomitant with the release of the catalytic subunit. We can drive it backwards, of course. Remember that our allosteric effectors, whether you consider cyclic AMP to be the allosteric effector or the catalytic subunit itself to be the allosteric effector, we can use one or the other to drive the conformational equilibrium back and forth. So in the presence of elevated concentration of cyclic AMP, the regulatory subunit dissociates, undergoes an enormous conformational change, and this is the reason for showing you this slide, is that this is a very similar notion to what we've seen in many other cases, that new chemical information, i.e. the binding of cyclic AMP, changes the structural information that's present so that we can populate a different part of this structural landscape, if you will. All right. Finally, something about this pseudosubstrate that I mentioned previously. So protein kinase A binds molecules and phosphorylates them, and the key is that into the regulation is that the regulatory subunit presents a pseudosubstrate, and the pseudosubstrate shares common structural features with the actual substrates. And that's sort of represented here in this cartoon. We see here's the regulatory subunit in gray, and its pseudosequence involves a couple of arginines that form a complicated salt bridge network with some conserved elements of the catalytic domains. So these are the binding determinants of not only pKa's phosphorylation targets, but also its its regulatory subunit as well. So I just want to again give you the idea that there's a structural basis for this regulatory activity, and it's this interaction that we disrupt when we bind cyclic AMP at the regulatory subunit. Okay, so finally, let's tackle tissue-specific activation of enzymes, or tissue-specific activity, that is to say. And this is not so much changing the function of a particular protein, but it's changing the overall function or the overall functionality by changing the presence of what changing the identity of the protein that's present. So the easiest way to think about this is during development we see a shift in the population of lactate dehydrogenase structures from one form to the other as a progress, in this case of rat, the development of the rat. So, first of all, definitions. What's an isoenzyme? Well, it's an enzyme. It's a pair of enzymes, for example, that have very similar activities, but they are encoded by different genes and have different sequences. In this case, of course, lactate dehydrogenase isoenzymes are very, very similar, perhaps better than 80% identity, so obviously related to one another, but they're under differential transcriptional control, and that control depends on the developmental state of the organism. So of course gene duplications arise, and they have varying activity and or expression patterns, which we see here. And in the case of LDH, there are at least two, sometimes there are more, but let's consider two versions of LDH in the developing rat. And in this case, we're looking at rat heart tissue, and these balls and squares, which I always run down, but I'm going to show you some balls and squares today. These balls and squares represent, on the one extreme, lactate dehydrogenase. This should be the M4, muscle 4 tetramer. So we have all the muscle version of lactate dehydrogenase, which does not respond to elevated concentrations of pyruvate. And we see as we develop that we switch to the H4, also known as LDH1, H4 version of pyruvate dehydrogenase. This is the heart version. So as the organism develops in its heart tissue, we see a switch from this more muscle version to that version of the enzyme, that isoenzyme that is more relevant to the aerobic conditions experienced in the heart. And and one detail here is that we see that elevated concentrations of pyruvate inhibit this molecule. And of course there are 5 versions of LDH that we get just by mixing and matching the two endpoints. And I think you see basically what's happening here is that there's a slow shift over the course of a few weeks to a predominantly H4 version of this protein. So again, this is reflecting a developmental change and the metabolic needs of the endpoint tissue. We've gone from a generally anaerobic version of the enzyme to an enzyme that's more optimized for functioning in aerobic tissues. Here is some, I was showing you some ancient data. This is the data on which that previous balls and squares figure was derived. This is development of lactate dehydrogenase in the rat heart. We see that in the fetus there is very little to no H4, that is to say LDH1, and we see that as we proceed to the adult, that we're predominantly the H4 version at the expense of the M4 version. That's the same thing again. Oh, and this was a fun slide that I added. Let me say that, again, this is from a very old experiment, but it persists in the literature, at least in the textbook representations of tissue-dependent modification of isozyme expression. Here on this axis we see 1, 2, 3, 4, 5, again referring to the 5 different versions of LDH that we see, remembering that these represent the 2 endpoints of the 2 LDH monomer types. And this is electrophoresis. So this is the sort of a cartoon of an electrophoresis experiment where the sample might be loaded here, and we see one version, LDH1, migrating towards one electrode in the electrophoresis, and the other version, LDH5, migrating towards the other electrode, again representing one of the differences between LDH A and LDH B that is, in this case, just having to do with the amount of charge on one or the other. But the key, again, is simply that after 6 days after fertilization, we see primarily LDH1, but then we see this switch happen, and in this case, it's looking like the switch really turns on after the organism hatches, and we see that in the adult, we have switched over entirely to LDH5. So again, that's just a third representation of the same experiment. And then again, finally I added this crazy Auger diffusion experiment, which I think I incorrectly characterized in lecture as an electrophoresis experiment. It's a diffusion experiment, and it's complicated, but it's just so crazy and interesting that it's worth talking about. In this case, what we're doing is placing our purified protein in spots here, here, and here, and looking at how they diffuse against antisera raised to, in this case, this is antisera raised against LDH1, and here's antisera raised against LDH5, and where they diffuse together, we see this band of precipitation. So we see that this makes perfect sense if we think about it this way. LDH5 is raised against antiserum 5 raised against it, therefore we know that there should be an interaction between these two, and in fact there is, and we see the duplicate here. Right, so this is just the precipitation of LDH5 by the antibodies raised in this antiserum. But the key here is that between LDH1 and this antibody here, we see some interaction, let's back up and say single line between LDH5 and the serum raised against LDH5 makes perfect sense. Another detail, though, is that between LDH5 and a serum raised against LDH1, we see no line. So we should see a line here if there was an interaction. Intriguingly, though, between LDH1, we see both interactions, of course, between its own antiserum. So this is antiserum raised against LDH1. We see a nice fat band. But we also see some banding between the antiserum raised against LDH5 and LDH1. So it's complicated. But the point of this slide, this whole slide, is simply to show that back in the 60s, what we're trying to define is the differences between these isoenzymes, and this sort of proves that LDH1 is distinct from LDH5 in terms of its ability to interact with specific antibodies. So they're distinct entities, which of course is much easier to demonstrate now. So I hope this was a fun lecture for you, and we'll continue our discussion of enzyme regulation in class.